Welcome guys. In this video we are going to look at two questions extracted from biology paper 2 for the year 2024 for the GCE. And for those that may be interested in joining my online classes, feel free to inbox in the comment section below. Right, so let's begin with the first question we have. Figure 1 shows a generalized animal cell. So if you look at a cell is considered to be a basic unit of a living organism, which simply means that the life of a living organism depends on a cell. Right, now we have two types of the cell. We have an animal cell and a plant cell, but we are focused on an animal cell, and we can tell from the shape that this is an animal cell because it is irregular in shape. Right, so let's have a look at the first question. So the first question is all about identify. So to identify simply means to state the name given to a labeled part. Right, so identify the parts labeled. We want A, B, and C. So A refers to this liquid here. We can see by the dotteds. Right, so this is called a cytoplasm. Then structure B, we are looking at the outer part of an animal cell. So this is a cell membrane. Then structure C, we have this small circle here. So this is a nucleus. So now we move on to question B. So question B, we are just told to name two organelles present only in plant cells. So this is same as the parts that we can only find in plant cells, but not in animal cells. Right, so here we can have chloroplast, apart from chloroplast, we may also have cellulose. Cellulose is a glucose kind of material that is found between the cell and the cell membrane, and cellulose is what forms the cell. Then also the cell is another structure that can be present in plant cells but not in animal cells. Now we move on to question C. We just want to explain the function of the part labeled B. So what is B? B is cell membrane. So cell membrane is the outer part of an animal cell. So it acts as a guard or a security, which simply means that it selects what to enter. It also selects what to leave the cell, because some materials may be harmful to the cell. So therefore they have to be guided when entering the cell. So say to select what to enter and leave the cell. Right, so we move on to question D. Describe the effect on the cell in figure 1 if it was placed in a hypertonic solution. Right, so let me give you a simple illustration. So we are going to have two beakers. Each beaker will have a solution, but the concentration will differ. So in beaker, let's call this one beaker A, we have this cell. We put this cell. Then also here we put the same animal cell, but here we are going to say this is 5% solution in terms of concentration. Then the cell is 10% concentration. Here the solution is 10% concentration. The cell is 5% concentration. So now when you are looking at this hypertonic solution, whatever solution that you are given, you need to bring in now the movement of water molecules by osmosis. So that's a concept that we are trying to bring in here. So what you need to know is water molecules will move where there is too much water into an area where there is less water molecules. So now how can you tell that this area has more water molecules compared to the other one. So you need to look at the percentage if you are given. So here it simply means that since there is 10% salt, it means that this solution in the cell is more concentrated, which simply means that there is more solute than the solvent. So therefore water will move from the solution into the cell, which simply means that the cell will gain water. So a solution where the cell gains water is called the hypotonic solution. So remember, hypo simply means below. It simply means that the concentration of the solution is less than the concentration of the cell. Therefore, the solution will lose water by osmosis into the cell. Then hyper. So hyper, according to the science, simply means that the solution is more concentrated. So since the solution is more concentrated, the water molecules will move from the cell into the solution. So the solution will now gain water. As the solution is gaining water, the cell will start to shrink until it reduces and changes its original size. So therefore, the answer should just be the cell will lose water into the solution by osmosis. And reduces in size. So which is we look at this one as shrinking. So this is what we can look at this question. So let's move on to question number 2. Question number 2 is about a rhizopus, and if you have looked at questions lately, this question has been coming almost every year. So figure 2 shows the structures of a fungus. So this is a rhizopus. We have structure D. So structure D is called the sporangium. So the sporangium is where the spores are released during the process of reproduction, which is, if we specify, asexual reproduction. Then structure E, which holds this and also which holds the sporangia, is what we call the sporangiophore. Then structure F, this horizontal, this one is called stolon. So don't confuse stolon to structure G. Structure G is called the rhizoid. So there is a difference. So let's have a look at the first question. So the first question is identify the parts labeled D and G. What's D? So D is sporangium, G is rhizoid. So we have sporangium. Then G, rhizoid. Then explain the functions of the parts labeled E and F. So what's E and F? So according to the structure, E is sporangiophore, F is stolon. So we just want to give the function of sporangiophore and the stolon. So E is sporangiophore. So function to hold the sporangia. Sporangia at their tips. Then F, stolons. So this to form rhizoids and sporangium. So to form rhizoids and sporangium. Right, so we move on to question C. Question C is under outline the process of nutrition carried out by the fungus. So the type of nutrition here is what we call saprophytic nutrition. Why is it called saprophytic? Because it's a type of nutrition where an organism depends on a dead body, something that is dead and decaying. So now, how do you outline this process? So we are going to say the fungus will secrete the extracellular digestive enzymes which will break the complex carbohydrates into simpler ones and absorbed by the rhizoid. So I've just tried my level best to summarize the answer, but the point is there is more to know about the type of nutrition under the fungus. So you just have to make sure that you study as your level best. You need to research more. So the reason why we also called the extracellular digestive enzymes, so these are enzymes that works on the outside of a cell. So that's what we call extra. So extra, outside, intra, inside. So you need to pay attention to that. Then for Roman number 2, give an example of a fungus that carries out the process of nutrition shown in figure 2. Give an example of a fungus. So Rhizopus is an example. So it can be Rhizopus, it can be mushroom, it can be muka. There are so many. So that's it for this video. Thanks for watching.